Hello, I'm gonna present the work that I've been trying to achieve recently. It's been 2 years that I'm working on this. My name is Adrien Lucca. I'm an artist. I live in Brussels, and I have this new passion for animal color vision. So my motivation is that so I discovered literature about this moth, and I became really interested by the idea that it may be possible to create pictures that represent how these moths see a flower. Um, and this comes together with an idea that I have, which is to create a form of art as an artist, which is somehow realistic because it's based upon the scientific method in order to portray things that are invisible to our senses. So I've already done a few works about this. This is an installation that is called the Blue Hour. It's an installation with real plants and LED lights, and it's an attempt at showing how flowers may look different in the eyes of a moth on one hand, and on the other hand, like what is the impact of artificial light at night on these colors that appear differently for them and to us. So this is what happens in the installation. The light is changing progressively, and you see, like for example, there's orange flowers that becomes blue and that finally becomes green, and the green is kind of the same hue as the plant. And so the idea is that an illuminant without UV, such as LED, may induce similar changes in color. I also made some video based on some hyperspectral pictures that I have created, but this will be explained in the second lecture. So the point of this lecture is mostly to discuss my main reference and the basis of all this research, which is a paper by Briolat and collaborators from 2021. And this paper has a very interesting framework. It has like a lot of ecological data. It has a quantal catch model and receptor noise limited model, contrast analysis, and it's asking some really good questions like what happens when, for example, a flower appears on a leaf under twilight versus how does it changes with, for example, LED light, low pressure sodium, etc. And this table is the main figure of the paper, and I'm going to talk about this. I think some of the methods and the results are actually highly questionable. The main thing that triggered this conclusion to me, I had a very strange first impression when I read the paper, because in the conclusions, it is said that white LED performs similarly to moonlight in terms of color contrast, or they may even enhance chromatic contrast between flower and green background for the moth, and this regardless of flower color. So I just want to point out here that an LED, a white LED, doesn't emit any UV. So because the animal is a trichromat and it's like photoreceptors, one of them is in the UV, it's very counterintuitive to think that LED lights may improve color contrast, while logically it should actually remove entirely one of the dimensions of this trichromatic color experience. So this was my starting point. The conclusions also say that this can happen with PC amber LED, which is even more weird because PC amber LED has no UV and no blue. This is compared to the moon, and they say that this can yield similar or even higher chromatic contrast than moonlight when the light level is high. Let's see about that. So this is the question actually, why these illuminants without UV seem to improve color vision in Briolat. So that is the methodology of Briolat. It starts with some data and conditions, then they compute catches, convert to XY space, and then compare. The first thing in the data is the natural and artificial lights. There are actually some little issues there. Some different instruments and wavelength ranges, different level of noise are present. For example, all the LEDs are cut entirely to zero below 400 nanometer. So I suppose they were measured with like a visible range instrument, while others are fully recordings over the 300 to 700 nanometer range, which covers the insect vision. The issue there is mostly that there are different level of noise in the different curves, and this is not really addressed properly, I think. Then there are reflectance like flower, grass, leaf, etc. This is very nice. There is a little problem that they categorize the flowers using human color categories. So like white, yellow, pink, purple. There's the moth body reflectance. Here, I think some specimens may have faded because in a paper by Johnson in 2006, there is a curve of the reflectance of the wing of that moth, and it shows like a very strong UV reflection, which is not in the Briolat data. And then there are like these like different illumination levels. These illumination levels are set to cover twilight to starlight. But if you read Kelber 2002, actually the lowest level that this moth has been shown to be able to discriminate between patches of blue colors is actually 10 times smaller. So then they compute the catches. Here there are many problems. So they use a bunch of parameters from the literature, but they use an I which is fixed, so there is no implementation of space and time summation in their model. But if you read Kelber, you have to assume that at low light levels, they will be summation, otherwise you cannot explain that they can see color at these light levels. But they also use like a 1 second integration time. They use a doubled Rabdom length without saying why. Actually, this is coming from Kelber and Johnson. The length is doubled because the light is reflected by the tapetum, so the absorption is computed like a second time. But they don't contextualize this. And finally, the catch equation has something very wrong. The catch equation itself looks pretty much like a standard one, but they use the functions from Johnson, which I call transfer functions. And these transfer functions are actually already the product of a Beer-Lambert absorption term, but they put that back into the Beer-Lambert absorption term. In between, they normalize it by area, which means that basically they apply Beer-Lambert twice, but with like a wrong normalization. And so there is a problem here that they use transfer function instead of pigment templates. They could have also completely dropped this term and use that directly, but they didn't. Finally, the 1 second integration time multiplies the catches by a lot, and this divides the catches by a lot, and you get somehow equal plus and minus. Then there's the XY conversion. Here again, there's a lot of problems. So they used the receptor noise limited as a color space, which is probably okay. But then they don't define a background. So the origin of their space is equal quantal catches. They use the Euclidean distance in that space to define the delta S formula between like a flower and a leaf, for example, which is different from the standard implementation. They use like a not really documented Weber fraction, but that looks very much like the fraction for a B. This is the most important. The noise model is computed differently than in other implementations. It's actually computed privately with the signal itself. But the problem is that the noise term in this XY representation is the metric of the color difference on the axis. So if you change the noise term for every sample, you're plotting samples that live in different definitions of the X and Y axis. And so with all of this, they then use like gamut areas and like delta S between samples in XY space to measure what they call color contrast. And so finally, they get to these comparisons in different kind of comparisons. This is one example. So these are like all data set of flowers and leaf in XY space that they put in their supplementary material. And we can see that regardless of the presence or absence of UV in the light, there's always like some kind of significant contrast in the y-axis. So all the LEDs here, the white LEDs, do not emit any UV, but they produce a UV contrast, which is of the same order of magnitude as like the moon, D65, starlight, etc. So that happens without any stimulation of the UV photoreceptors, or like with a very, very tiny stimulation. It's also important that actually they put a patch This is a kind of rough, very rough filter on the light spectra. They never talk about this in their paper, but it's in the code. The idea is basically that they will first clean the emission spectra by removing all negative value and setting them up to 0. And once they have done this, they take all the values that are below 1% of the maximum value and they set it to 10 power of minus 16. Why they do that? Well, probably because if they don't, their model actually predicts like very large areas in XY space for the low pressure sodium lamp. So with the patch, the area is 0, and without the patch, the area is about 200. The only difference here is the amount of noise here in the curve. So that curve is unpatched, and that curve is patched. So this one is noisy. We don't see it in a linear plot like this. And this one has like a very flat baseline at 10 power of minus 16. So this prediction is obviously wrong. Low pressure sodium, like a clearly monochromatic illuminant, doesn't produce important color contrasts in XY space. There is something very weird here. And so I basically lost trust in this paper that I wanted originally to use to try to model how the insect is seeing flowers. And instead, I implemented from scratch a completely new photon catch model that includes like a new rhabdome model, and it also includes like the pupil and the temporal and spatial summation. So this is the M4 model. Then I added like a parameter to the receptor noise limited model itself because I found that this was probably necessary. This will be discussed here. And I also created a hyperspectral rendering pipeline in order to be able to somehow see through this simulated optical neural system of the moth. But I'm not going to present that hyperspectral picture in this lecture, but in the next one. So what is the M4 model and how does it compare to the Briolat quantal catch model? So it's an absolute quantal catch model. It starts in the sky with ecological conditions and on the ground with reflectance. Then it goes through the pupil, the rhabdome, and the summation. So it starts with more ecological conditions than Briolat because I added like a full twilight sequence. Then I implemented like adapting optical parameters and neural and spatial and temporal summation that I took from Anna Stoker. And like this, we get noise-weighted receptor chromaticity contrasts with a parameter for spectral support that I will explain. So first, the ecological conditions. Well, I took like a data set from Spichan and collaborators 2016. This is very nice because it presents like a full twilight sequence from sunset to the starlight level without light pollution because it was measured somewhere in the US where we are far from any kind of city. And the data set also contains the moon or not, and it allows like modeling the radiance, the absolute radiance of the sky in function of the solar elevation. And with this model, I noticed something interesting that, for example, in Brussels, where I live, on the summer solstice, the sky is actually blue the whole night. And so this is, I think it's a period which is important for the moth, and it's also important in terms of like how do we imagine the night and how do we imagine what light pollution is, because light, artificial light exists, pollutes that kind of light. It doesn't pollute a black sky, it pollutes a blue sky, even if we don't see it. So these are some frames of that data set. So it starts with daylight, and then like there's less and less green light, and then it is a kind of it's taken over by starlight, air glow. And it's interesting, important to check like there's UV in every frame in about the same proportion. So for the reflectance, flower, and leaf, I took actually the data from Briolat. I also have other data sets, and I also measured some things by myself. So next is the eye adaptation. It starts with the optical part, so the pseudo pupil and the acceptance angle. And then so these are like the graphs from Anna Stockel. And then there's also like spatial and temporal summation. So all these things have been measured by Anna Stockel, as far as I know, with the same calibrated computer screen and a real mouse, and or I guess like several real mouse. And importantly, the screen has no UV emission. It's a computer screen for human beings. But in a paper by Hamdorf here in 1986, there is this observation that the pupil, so the screening pigments, the pupil reacts mostly to blue and UV light. So I assumed that maybe in these figures, what was triggering the reaction of the pupil was maybe just a very small blue part of the emission spectrum of the screen. This may be true or not. This is how I modeled this, and I think it could easily be verified if this is right, if this is overestimated, etc. So I end up with these 4 parameters for pupil state, summation aperture, optical acceptance angle, and temporal summation gain. So these are like 4 Stockel derived parameters, and they define like the eye state in my model in function of the current observation conditions. So then after that step, there's the eye anatomy of the rhabdome itself. So I checked a bit in like classical references in the literature, and I ended up thinking that it may be interesting to rework the model that was developed by Johnson, which Briolat is using, because that model by Johnson basically models the rhabdome as a kind of homogeneous pigment soup within a volume with a tapetal mirror behind. But in fact, in the literature, we can kind of deduce that, well, first of all, there are these 2 rhabdome types, and they are structured. So like the blue or UV absorbers come first, and under is like a green absorption zone, and there are different volumes. So there are only 2 photoreceptor cells for blue or UV, but they take a big part of the rhabdome volume in the distal area. So I modeled this like this as an average rhabdome of the 2 types, with a distal area where there's like a ultraviolet and blue absorbing quarters and 1/2 of green absorption. And in the proximal, there's a full green absorption, and there's also a mirror tapetum full reflection that is implemented. So the photon first pass like this, they're screened, and then they are reflected back. This was actually already modeled by Kelber and Johnson, but with like different volumes. So it looks like this in 3D. It's like a kind of cylinder where we assume that the photons are only entering in the axis of the rhabdome. In order to create the transfer functions of this rhabdome, I used SSH pigment templates exactly the same as Johnson, because everything is in his annex. And this is the result. So this is the result of like a Beer-Lambert absorption system, which is defined in Johnson. These are what I call unnormalized transfer functions. So this is what that part of the quantum catch equation creates. And we can compare here on the left with normalized transfer functions that are also created with the model of Johnson, which is an equal fraction rhabdome, meaning there are the same volumes of like violet, blue, and green absorption volumes. And then it's renormalized such as each curve has the same area under the curve, and then the whole thing peaks to 1 with UV peaking to 1 here. This is actually designed like this because the goal was to plot colors in Maxwell triangles. And when you plot in a Maxwell triangle, it's really nice to have equal areas under the curve, because when you have like equal quantum flux at every wavelength, you end up with a point right in the middle of the Maxwell triangle. But this is not what we are trying to do, so we use the unnormalized function, and we can always renormalize them if we want to plot in a Maxwell triangle. Just a reminder, like Briolat is using this within that term, and in fact, this is already the product of that term. So then we have the catch equation. I tried to make it very simple. So we have like a prefactor, an integral of photon radiance, and the transfer functions. That's it. Then the prefactor itself is explained more in detail here. It's nothing special, all classical parameters that everybody's using. And then there's the photon model, the photon noise model. I think this is quite important. It clarifies a lot of things. So the catch equation can create two kinds of catches. The expected catches, which are the red line here on the graph. But we can also compute realized catches with the Poisson process, and that will simulate counts per integration time. And we see that the counts are actually like not always the same, but they form like a bell shape around the mean, and the mean of the counts is equal to the expected catches. But these are two different things, because the expected catches are floating values, but in reality, you never have like a floating point photon which is absorbed in an integration frame. You always have integers, like you cannot divide a photon in 0.3. So in order to try to model that like rigorously with the questions of spatial and temporal summation, I implemented like a random number generators that has several temporal seeds that create several frames. And in these frames, we have like different rhabdomes. And so each rhabdome creates its own like unique Poisson process to generate counts. And then you apply temporal summation and spatial summation within these frames at the same time, and you get like this NT, which is the sum of the temporal frames integrated by the summation spatial summation window. And so now we can see what we get. So this is a Maxwell triangle, and in there we have Poisson clouds. So I call this a Maxwell-Poisson color triangle. It's pretty nice because we can see where a flower is landing within the insect chromaticity space. So the channels are like, the vertices are the primary color, UV blue-green, and we have the spectrum locus here, which is the place where the pure wavelengths are. So actually this creates like a smaller triangle within the triangle, which is the 2D space where any kind of tri-stimulus can live. And so when we zoom, we see these Poisson clouds. Some are like smaller, some are larger. What changes the sizes here is basically that some leaves are much darker than the flowers. The signal is less important, and so it makes like larger clouds. And when we dim the light, every cloud is getting larger. So this is one kind of representation that is interesting, I think, because we get to know the chromaticity between the different flowers and leaves. And also we get like an intuitive idea of how noisy that is at a certain light level. So the main result of that is that the eye seems like extremely well adapted to twilight. These are like three figures that correspond to the number of catches in every channel, green, blue, and ultraviolet, that are produced across the full twilight sequence of speed-chan, so between like sunset and like starlight. You have the luminance here, which is written as an indicator on the top. And here it is rendered as if the animal is looking at a Lambertian pure white diffusor. So it's a little bit like the situation where Anastotle was putting insect in front of a screen. And there are three figures because the first one represents like a fixed eye with a pupil that is always open, no summation, no adaptation of any kind. So this is very close to what Briolat has used. In the middle, we see immediately that adding the pupil, aperture, and closure creates like a local maximum somewhere here in the twilight. So it's interesting because we break this rule that more light is always more catches. In this zone, it's not true actually. If you put more light, pupil is going to close faster and you will get less catches according to the data of Stockel. And here we have added the spatial and temporal summation, and the result is that the dynamic of the amount of catches across the full sequence is much smaller than with like a fixed eye model. And also the amount of catches at starlight level is still very significant. So this is a zoom, and we have these two local maxima here, and we see that even this one, when the sun is about like minus 14 degrees, which is like the maximum of catches that is within the rise of the spatial temporal summation, we still get 25,000 catches in the green channel, which is really far from Briolat values. And we can also input like something else than this Lambertian white diffusor. So here, for example, I anchor the system on a green background, and I plot the catches produced by a flower within these conditions. And this gives like surprisingly high values here, around like 100, a bit more than 100 candela per square meter. We have 8.5 million photons in one photoreceptor, uh, which seems much, but also like it's interesting because this is the moment where these flowers, Enothera is open just after sunset, and so I'm not sure if this is totally correct, but it's an interesting result. So the first big difference with Briolat is the magnitude of the catches, especially at low light. So in this small table here, we have 1 candela per square meter versus 1,000 of a candela. So for Briolat, we have like significant values at 1 candela, but still like for the green, 32 times smaller than ours. And at very low candela, we arrive in this like 11, 20 kind of photon values for Briolat, which with which it would be very extremely complicated to explain any kind of color perception, while with our model that includes the summation, we arrive to 1,000, 2,000, 20,000. So for the flower. And so these values, like I think, can support the idea of color vision at that kind of light level. The second part here is for the leaf, the background, and the background has much lower values, but still significant, probably for us. Like here, it's becoming really small, like 40, 49. But for Briolat, it's basically nothing. Like it's 0, 1, and 3. With 0, 1, and 3 photons, I don't think you can trust what like the next step will try to predict. So the next step is the receptor noise limited model itself. I will compare like different versions. I would say the Kelber, Vorobiev, and Osorio version is the classical one. Also introduced some kind of modification. Then there's the Briolat version, which is really unique. And then there is our version that we have modified a little bit, but which is mostly Kelber actually. So in the receptor noise limited model, there is a metric. We can write it like delta S. It's a metric of the full receptor contrasts, and it can be like represented in 2D in an XY space. So the chromatic discrimination delta S is fundamentally limited by the receptor noise E, therefore the name of the model. We are talking only about chromatic contrast, which means that any variation which is proportional between the catches, which I call common mode or achromatic variation, is completely disregarded. An N receptor system has N minus 1 dimensions, so UV blue green becomes XY 2D. It's a bit like a chromaticity diagram for human beings. And I think that something which is not addressed, but which should be true, is that there is an implicit requirement of using broadband light sources for this model, because we will see that in the way this model computes contrast, it removes like the relative signal amplitudes in the log contrasts, and it also cancels like any kind of catch ratio, for example, how many UV against how many green photons are in the catch signal. It removes that entirely from the noise definition. So I compared the four models here. I'm not going to explain this fully, but the important part is that Briolat is really an outsider here. It doesn't use log contrasts. It uses the log catches directly. The way it models the noise is completely different from the others because it uses only the noise of the sample that you are actually considering um, to compute the noise, which means that the metric is fundamentally different from the others because each point has its own X and Y axis by definition, because it's the noise that creates the metric of the axis. They don't define a background, so they are the only one that cannot apply the classical delta S formula because it requires a background. So instead, they compute Euclidean distances in XY plots. And they also get like very different numerical results, and we're going to see this. So an important part is the Weber fraction. That is the main contributor to noise in this model. It's basically, the idea is like how much you should change some kind of signal in order that it becomes something that you perceive. It comes from psychophysics, and it's at the heart of the receptor noise limited model. And there are different conventions on how to which letter to use and to what it refers to. So I want to say Briolat used like a Weber fraction of 0.05, but it actually corresponds to a Weber fraction of 0.13 in another convention, where we made sure that we are using exactly the same Weber fraction as them. The Weber-Poisson transition is like, yeah, so this is a representation of these like transitions between pure Poisson, pure Weber, and like the kind of intermediary zone where we may be at very low light in some kind of pretty graphs. And here we can see how it changes if you change the Weber fraction. This is the noise equation. So the noise E is the square root of the Weber fraction squared plus the inverse of a quantal catches, which is the reference for the noise calculation. So usually when you plot XY spaces, you take the mean of the background and no, sorry. When you plot XY spaces, you take the background catch. When you're just comparing pairwise like flower and leaf, you can take the mean of the background and the signal. So then the noise sits right in the middle of the two samples that you try to evaluate in terms of like chromatic contrast. And there is one value which is interesting, that is n star, I call it. It's the inverse of the Weber fraction squared, and this is like a value that can tell you at which number of photons you're passing in the Poisson photon shot noise regime, or in the Weber normal noise regime, where adding more signal is not increasing the quality of your discrimination of differences. So the normal Kelber style way of computing the coordinates is like first taking the log ratio of like a stimulus in the background and computing the noise with a reference that is fixed. That can be either the mean of these two or a single value. If you want to plot a bunch of value, you need a single one. And then you project that on X and Y axis. I will show it after. So I said that like the XY projection in that space requires a single noise anchor. This is actually very important. So the Briolat is computing the noise like this. So they take like, they say that the noise is the square root of Weber square fraction squared plus the inverse of the signal itself. So then they compute, for example, like how it ends up to be an X coordinate, but this X coordinate is like pre-computed using the noise that uses the signal itself, which means that every time you're doing this, you're using a different scale. And so in their work, it works like this. You start with the absolute catch, you create the noise based on the absolute catch and the Weber fraction, and then you project in XY. And that creates like a strange behavior. So this is what I'm going to show here. The plot here on the right represents what happens when you start with equal quantal catches, so 1000 everywhere in UV blue green. And then we divide the blue and green catches by 2 every time until we arrive to a very small value. So for example, the last one here is like 0.03 in UV in blue and 1000 in green. Logically, that should be like a very saturated color because it's like pure green. And when you put that in the Briolat noise model, actually the noise is increasing so much in UV and blue that it's crushing the coordinates of X and Y. And what happens is that you have like a signal that is first treated as being more and more chromatic, but then it goes back to the origin as if it were becoming achromatic. While in the classical XY color space, because you have like a fixed noise, then your noise is not changing. And what happens is like these are like equivalent to like logarithmic equal distances, and you get indeed equal distances in that log space that is the receptor noise limited representation. So the Maxwell plus RNL plus Poisson is a kind of nice diagnostic tool, I think, because in the RNL you have the J and D distances. The Poisson is a kind of nice, the Maxwell is a kind of nice way to always check where your signal is. It never moves. And the Poisson clouds tell you about like how noisy that thing is. I just compare here Briolat versus the Calver version of the same samples, and we see that in Briolat the things are moving around like in kind of unpredictable ways. The origin is also very far from the figure because it's equal photon catches in every channel. So we always like very negative and positive in X. Here the background is defined, so this is the origin of the figure. So we can test now, like for example, let's put an LED in there. We have the spectrum here. We have in photon flux, we have like the realized and expected UV catch here that's extremely small. In the Maxwell triangle, everything is collapsing to a single line. So it's like we pass from a kind of 2D cloud of data to a 1D line of data. And in the Kelber receptor noise limited XY space, we see that the clouds are like very large, but all the data aligns also on a single line, a bit like in the Maxwell triangle. If we put that into Briolat now, we have like a completely different story with like very large contrast on the y-axis, and the contrast on the y-axis are actually UV contrasts. So the Briolat receptor noise limited convention or conversion actually predicts large contrast with UV-free illuminants, such as LED. But there is more. Actually, the receptor noise limited is also not perfect. So I'm just gonna make a little break here and ask you to imagine what is actually the act of perceiving the color of an object. So perceiving the color of an object is not something which is directly related to counting the photons that reach the photoreceptor in the eye. It's actually an action where we try to estimate an objective property of the object that we are looking at. We call that sometimes its color, but in this case, it would be maybe better to call it the reflectance of the object. So the color is maybe not like an objective property, but the reflectance definitely is. And the illuminant and the photoreceptors are like kind of talking together, and the illuminant is the probe that reveals what the reflectance is, and that is perceived eventually as a color. The important part here is that an illuminant, if it's broadband, it will scan the entire reflectance. But if it's not broadband, then it may not reveal the full reflectance, and then the object can have a completely different color. So this is very well known with low pressure sodium. With low pressure sodium, you get this very monochromatic world. So all the colors here can only be like I absorb or I reflect a yellow photon. And so you have only shades of black, grayish yellow, or brighter yellow on the picture. But this can also be done like with white light, like this is my work. For example, here the illuminant is in the back of the room is a mixture of turquoise and red light. And when the balloons that are yellow under broadband light arrive in this space, they become red because there is no green in the light. And so we are unable to probe the reflectance of the object. This is one of the ways to describe what is happening here. So what's happening if we put the low pressure sodium in the receptor noise limited model? So in the Maxwell triangle, what happens is very simple and logical. Everything collapses to the green corner because most of the catches are there. But in the receptor noise limited model, the picture is different. So in Briolat, you have like huge contrast in low pressure sodium from the file that they provide. But also in Kelber, you still have 15 units of contrast in X here, which is also huge actually for an illuminant that supposedly turns everything monochromatic. Interestingly, we see in the Poisson clouds these little jumps here, and these jumps are actually one single photon difference, which would mean here sometimes that the one photon difference creates chromatic difference, which would be perceived as such, which is difficult to believe. And the PCM light behaves in the same way, so you have like a almost complete collapse in Maxwell and like a large expansion in even in the Kelber XY space. And in the Briolat space, it's totally gigantic, which is maybe why they say that PC Amber sometimes outperform moonlight. So we want to understand what happens here, because this is really not logical that like a monochromatic illuminant is predicted by a color discrimination model to create like important color contrasts. So under low pressure sodium, only the green channel receives some information, and the UV and blue essentially receive 0. But in fact, the file, the recording, the illuminant itself is noisy. So there is some kind of signal in the file. As it would be in real life, there would be like a low pressure sodium lamp somewhere and a little bit of twilight or a little bit of other light sources. And we would end up with the same kind of information where we have like very, very little photons everywhere and a huge signal somewhere, which is low pressure sodium light. The problem is that the receptor noise limited model is unable to discriminate between what is noise here and what is the true signal. And I don't think that the visual system does that. I think a visual system does discriminate like between what is background noise and what is true signal. So we can show this with PCM, for example, that is identical in its behavior to LPS. We see that the spectral curve is very noisy. And now if I cut the UV part, then the expected photon UV becomes like almost 0. And suddenly it realigns in a 1D line like the white LED before. So here, this only to cut noise is actually driving a clear artifact of coordinate expansion on the UV channel. So I remove the UV, which is only noise, and suddenly the y-axis here is collapsing entirely. This is true in the Kelber version of the receptor noise limited model, but not in the Briolat, where even cutting UV keeps creating like huge areas like this. So we actually have two artifacts at once. We have ones for the first one is like a specific UV expansion that is present in the Briolat implementation, but the second one is another still expanded XY representation that exists also in Kelber, Vorobiev, and Osorio version. And what explains basically that before the LED was aligning in a 1D line is that in the Briolat dataset, the LEDs have no information, the UV at all. So this is the main driver of the figure, in fact. The fact that here there's no noise, here there's noise, that creates a huge difference in this model. So we want to understand why. So here we compare like the moon and low pressure sodium. So we have the catches here. We see that like the UV catch, for example, here represent like 0.44% of the green catches. So this is like a very, very small signal overall. But then these values are still used to compute like the noise term of the equation. And here, I mean, it is justified because the noise term will increase. And so it's supposed to break down like this kind of signals. But we see that the increase in UV is only 34% with this model. Between like Moon and LPS, the noise value is actually not that different. And then third, we compute like these log ratios between the flower and the leaf. And we see that these ratios, they don't really care if they are calculated with this kind of residual noise things. Like here, for example, 800 by 70 is 2.5, or with like a proper signal like in the moon, where you also get 2.5 for the same reflectance. It's basically a representation of reflectance, which is almost entirely independent from the illuminant. And then you divide one by the other, and you get like this delta in a UV shortwave in green, and then the total gives you like the chromatic contrast. And here we see that even with this system that is purely a classical Kelber receptor noise limited pipeline, Enothera flower is predicted to be more discriminable under low pressure sodium than under the moon. So I think like there are some problems here. So one is that this noise denominator has no information about the scale that creates the noise, because here it's like 0.44% of the green, but it could be 10% like in the moon. It doesn't change, it doesn't matter. Like the noise is computed without that kind of scale information. And then the log ratio also cancel any kind of scale. As I said before, they're basically just a representation of something related to reflectance. So the log of 37K over 3000 is kind of the same as the log of 800 over 70. But one is like produced by a ratio between two extremely noisy things, while the other is produced by a ratio between two like proper signal. So the quality of these things is very different, and it's not explicit at all in the model. So we have two scale-free problem, like we have the log ratios that erases the signal magnitude, and we have that the static noise model that doesn't care about like what kind of signal is that reference here. And I think that this will be fine if the illuminant are broadband, but this is very problematic if the illuminant like LPS is actually creating a huge signal in a single photoreceptor class and a very small signal in the others. So I think that this model actually implicitly requires broadband lights. So can we fix that? I think so. I think the problem is that it's mostly in the noise term. I think it's the best place to introduce something. So what we do is that we add a small parameter called SI. And this parameter, the function is to downweight like signals that are very unimportant because one is highly dominating, and it can also upweight some very strong signals. So basically we pass from this first formula to the third one, where we divide the classical formula by the square root of that S parameter. So let's take an example. Like we have PC Amber here, an illuminator that is very biased, and so everything collapses in Maxwell in one corner. We see like here the photon counts. So the photon counts in blue and UV are very small, and the photon counts in green are like very high. Here you have like the states, I adaptation states of my model. And when we put that in Calber, we get this with these colors that are false colors, but that represent actually chromatic contrasts. And we see that in Calber, we have this large color contrast with these like very little counts of a PC amber, which is mostly noise. So we have two options to correct this. One is like an ecological model. We will compute S eco from actually a measurement of how much the surface reflectance variation by all the illuminants of the data set create like a panel of colors. So we train like the parameters on the Spitzchen and Briolat data sets with twilight, flower, and leaf, and then we measure standard variation in this world of like illuminant times reflectance. The parameter can be between 0 and 1. It can actually be above 1. It depends how we design it. So when we apply this parameter with like PC Amber LED, we get a support that is completely different. We get like 0.02, 0.02, and 0.56. And that is crashing the coordinates because we have changed the noise function in the model. And so we also see, even on my false colors, that everything has become much more achromatic. And we see the Kelber X and Y values here are actually very small. We are between -1 and 1. So very small units, almost impossible to distinguish between these colors now. Another way to create that parameter is without an ecological data set. We can basically estimate how much the illuminant will favor certain channels over others, which is an indirect way of recovering the ratios, and it acts more like a kind of spectral noise filter. So the idea is to compare how much photon are supporting a channel over how many photons are supporting the best channel. And we can do that in like a naive way, in a linear way, but we can also implement this in a bit more elegant way with a nonlinear approach. And the result is extremely similar. So here the supports are now like 0.2, 0.3, and 1. This one always normalizes the best channel to 1 by construction. And the plot is also achromatic, and chromatic distances here for low pressure, no, for like PC amber are very small. So we can compare, like here we have twilight. We see the three models, like the Kelber is the middle, the last one we have seen is compressing a little bit the twilight compared to Kelber, and the eco is expanding it a little bit. But the big difference happened when we introduced like PC amber or LPS, because in Kelber we have huge expansion of the LPS space that shouldn't be real. But in our two versions, we have a dramatic compression, which is very much like what happens when you experience like monochromatic luminance. And it's interesting to see that at dimmer light levels, this artifact actually disappears in Kelber, and I think it's just because like the noise in the spectrum of LPS is passing below or above this N star value that I mentioned before, where we cross into the Weber regime, and so noise can be interpreted as signal by the model. So I tried to make a nice approach to false colors by like implementing two ideas. So one idea is like how much the color constancy is preserved, meaning like you have object of a certain color and they don't, and your perception of the color of these objects is not changing if the illuminant is changing. And the other is chromatic adaptation, which is like how neutral the illuminant in which I am currently is. And I think the spectral support allows like to represent that pretty well. So for example, here I apply it to the LPS. So the background is now like this kind of chromatic. It's not gray anymore. And in Kelber, we see that there are clearly different colors here against that greenish background. And when we apply our parameter, we recover this chromatic experience, which is achromatic, but is not, doesn't mean that the illuminant itself has no color. A last thing I want to mention is that I tried to check how correlated in the different models are the log quantal catches versus the XY receptor noise limited coordinates. And it's interesting to see that in Kelber and in our two different parameter additions, the correlation is clearly perfect, but in Briolat, it's not at all correlated. So that means that there is something going on where between the log ratio of the quantal, between the log of the quantal catches and the XY representation, there's something completely bizarre going on, which in fact is this idea that the noise is defined only relatively to the signal without any background. So now we can retest the questions of Briolat. I need to talk about this figure because I tried to reproduce it. I studied the code really in depth to understand exactly what this figure was doing, and I ended up thinking that this figure is actually not the product of a code, because there are some parts in the figure A here, the first column, that are impossible to reproduce exactly. So we don't know actually which spectra they mean when they say white LEDs. For the other, it's easy because there's only one spectrum in the data set. And also for PC amber, we don't know because there are also different PC amber LEDs in the data set. And none of them is really truly matching the results here. The biggest difference is for white LEDs. So flower and leaf contrast white LEDs. We have never been able within their data set to get this all green kind of cell. There's always in the Briolat model itself some kind of like orange, red going on here on the right. I'm not sure if I got something wrong here, but it was impossible to reproduce that figure, that's for sure. And the information is not sufficient, and the code shows it. In the code, they say that they have to compute something and then make it outside of R, so outside of the software, meaning probably by hand. But I still try to compute that and like compare Briolat version that I could reproduce with my modifications, and the picture is changing a lot. Like there are not so many improved green zones anymore, especially LED systematically destroys the top part, which means the white and yellow flowers. And so the picture is very different. And so I can also like replot their material in their space. So this is the re-implementation of a plot that they have in their supplementary material. And we see that what's remarkable is that there's only 2D plots or 0D plots. So every luminance is creating a kind of 2D space here at 10 candela per square meter, except for LED orange and LPS, which end up in a single point. In our implementation, the picture is very different. Like the broadband illuminants have 2D representation. The dual band have a 1D representation, which makes sense because, for example, in an LED, if you have only blue and green light for the moth and no UV, you have a bichromatic contrast, which ends up making a straight line in a color space and not a 2D cloud. And then we have the 0D LED orange and LPS, which are same result but computed differently. So we have like very different conclusions here. This is something that we have done that Briolat didn't do because I think because their model doesn't work well when they do this. So we are mixing here a certain twilight frame with different sources of light pollution. So here we have like the twilight at 1 candela per square meter, and here we add a street light, LED, PC amber, so white LED, PC amber, and LPS at 0.25, 1, 4, and 16 lux. 16 lux is a value that I measured under a street light here in Brussels, so I think that it's the right order of magnitude. And we see that when the twilight is at 1 candela per square meter, the effect of light pollution is very minor. There is some compression here, but at 16 lux, but it's not a huge effect. When we divide by 10, we start to see like a strong compression at the higher 4 and 16 lux light pollution levels. And when we divide again, then we see that these light sources are completely crashing, dominating the color space. And interestingly, if we go even lower, we get these expansion effects, but that are purely driven by the difference of light intensity here. The gamut is a bit larger when there's a lot of light in this model. If we do that with Briolat, we see that the model makes truly absurd kind of predictions. So here we have like a systematic increase of gamut when we add light pollution, meaning like in every case here, whichever LED, white, amber, or LPS, this is always increasing the contrasts when we mix twilight and artificial light. When we divide, we get this continuous effect where like LPS, for example, starts by increasing the gamut, then increasing it even more, then even more, and then suddenly becoming 0. And when we go even lower, then we get like things that are probably meaningless entirely because we are speaking here in the Briolat model of sometimes fractions of a photon per photoreceptor per second. So we think that what we have tried and started to implement is better because we can actually see how the illuminance, how light pollution has a dynamic effect on the color space. So like here we have different kind of artificial lights that are mixed with twilight, and we see, okay, the space is reduced, the colors are shifting, the 2D becomes 1D, so all these effects, or 0D, so all these effects can be like seen dynamically, and we think that this is a much more faithful representation of what's going on. So to conclude, the answer of the question like why UV-free luminance appear to improve vision in Briolat is first the private noise issue because it inflates the y-axis systematically. Then there's the patch, where they replace like low value with even lower value. This actually sometimes amplifies this effect in a counterintuitive way. Then the model itself is designed for broadband lights, so it doesn't work well with these discrete lights such as like LED and LPS. The log ratio and the noise model cancel scale, and this ends up making these noise as signal effects. And also, finally, color discrimination in Briolat is defined as a 1T contrast, which is kind of a huge simplification, because the question, instead of being like, is the contrast changing? The better question, I think, would be, is the color changing? And that is a very different question that cannot be answered by a measurement of contrast. You can have like a constant contrast between two samples with a complete change of the color of the two samples. So if you only measure contrast in one dimension, you're completely blind to what artificial light may have in terms of effects on color perception.